Okay. So, hi all. I'm very pleased to welcome Dr. Sushmita Paul, who is currently an assistant professor at IIT Jodhpur for the IBSE Seminar. So, she has a BSc from University of Rajasthan and an MSc from Banasili Vidyapeeth in bioinformatics in 2007. And she finished her PhD in 2014 at the University of Calcutta, Indian Statistical Institute. And her research areas are basically in pattern recognition, in which she's even published a very interesting book on scalable pattern recognition algorithms. And she also works on microarray data analysis and omic data analysis in general. And she has, you can look through her webpage. She has like many interesting publications which cover a variety of areas, but looking at, microRNAs, omics data, and things like that. And I think you already have had a good interaction with her regarding feature selection and so on. So today her talk is going to be on identifying cancer subtypes using a feature weighting approach. So let's hear it from Sushmita. Thanks, Sushmita, for stepping in, and hope things are all going well there. You must have heard all the weird things happening at IIT Madras, and I hope our campus also gets back to normalcy
Yeah,
soon.
yeah. Thanks a lot, Dr. Karthik. I'm really glad to present my work and interacting with you and the great minds at your center. So thanks for this great opportunity to me. So having a great interaction with you people. So yeah, so without taking much time of yours, I will start presenting my work. So I hope you can see my screen.
Yes, yes.
So today's talk is the title is identification of cervical cancer subtype using feature weighing approach. So before going into the algorithmic part and all, and just few minutes back, I have also interacted with the students, giving an idea that what is cancer subtype and all means. So in personalized medicine specifically or precision medicine, we know that for a particular kind of cancer, let's say it's a breast cancer. So if I give a generic medicine to the patient, then the patient may not respond appropriately or the way it is thought of. So it is very important to know that what kind of subtype that patient is suffering from. So there can be different types of cancer subtypes. So it can be luminal A, luminal B, basal, and then there is a Claudin low as well, right? So it is very important to distinguish these types of cancer subtypes from each other and come up with a responsive biomarkers of these type of cancer subtypes so that appropriate prognosis can be done with a much more effect to the patient, right? So in this regard, several computational approaches have been developed. So, and as I mentioned in my interaction session also, that instead of having a single type of data, it is always advisable that we should be going with different types of data types so that we get a full picture from the for the problem and from the data. So earlier, what was practiced here in last 1 decade, it was practiced using, so if I want to identify subtypes of cancer, then let's say only gene expression data was used. Just a minute, I will put on a slideshow mode. I just forgot to do that. So here, I hope you can see my screen.
Yeah, yeah, yeah.
So earlier in last 1 decade, what was the practice is that with the advancement of technology, gene expression data was pretty much available, and then just simply using a clustering algorithm, one can get cancer subtype. Then later, what happened is that multi-omics data came with the advancement of technology again. Different types of data types were generated and they are deposited in public repositories like TCGA, ICGC, right? Now, what was the practice here is that for identification of cancer subtypes is that on individual data type, there will be clustering, and then there will be a weighted average or weighted step will be taken up on those clusters, and ultimately the clusters will be integrated, and at the last step, and then cancer subtype will be identified. Now, the third approach is data integration. Now, what happens is that rather than doing individual clustering on individual data types, first the matrices are merged into a single matrix, and then on that merged single matrix, clustering is done and cancer subtype is identified. Now, why first question can be coming into your mind that why I should be first of all going from a single mode type of data or single omic data to multiple omic data. That I have already demonstrated in my interaction session that in a biological process, it is not a single gene or single microRNA that is causing the dysregulation of that biological process. It is a combination of different types of biomarkers that is actually responsible for the dysregulation of that biological process. Now, we have to choose the data types very appropriately or carefully so that they are having some kind of connection between them at the level of biology or the molecular mechanism, at the molecular level. Now, for example, if I choose gene expression data and microRNA expression data for developing an algorithm, I know from biological point of view there is a negative correlation between these two type of data. Now, in a biological process, if I take a molecular or regulatory module, so I know that there will be a transcription factor, there will be a microRNA, there will be a gene. Now, transcription factor is helping expression of the gene, and then if a microRNA is there, that once the gene is expressed, then the microRNA, if it is present, it will lower down the expression of that gene. So the regulation of that biological process is dependent on so many different type of biomarkers. Now, if I only choose gene expression data, I may be getting the transcription factor information from the gene expression data. Now, I will be able to see that, okay, there are certain genes which are dysregulated, although the transcription factor is not dysregulated, or it is upregulated, and the gene expression is getting downregulated. Then what is the intermediate step that is causing that lower down of the expression of that gene? It could be the microRNA. Now, if I merge this information, I will get a full picture out of it. So that is why I should be going with the multi-omics approaches. Now, so that was a biological angle that why I should be choosing a multi-omics data. Now, the second problem for merging this kind of multi-omics data and identifying cancer subtypes is data integration. Now, if I simply, so in the second step, you see that I will be doing, so the consensus clustering, now what is this kind of approach is that on individual matrix, individual clustering algorithm will be implemented, and later the clusters will be integrated. Now, if I do this kind of approach, what will happen is that I am not able to see the relationship between the gene expression and microRNA expression. I am just doing independent analysis. At the end, I am just merging. So again, the biological knowledge is not fully utilized. So there is a high requirement for going into data integration type of approaches. Now, data integration can be facing another kind of challenge. So in a gene expression data, the variance can be different, and in microRNA expression data, the variance can be different. So I have to choose very carefully that how I should be integrating these two types of data. So the approach for data integration is that I have two matrices, and I will be merging these two matrices into one matrix, and then I will be getting, ultimately, I will be implementing clustering algorithm on that and identify the subtype of cancer. Now, I have actually, Madhumita and I developed an algorithm that got recently accepted in the IEEE transaction on computational biology and bioinformatics. And I will be expressing that how we have developed this algorithm, what was the computational angle in the algorithm. So here, in next slide, I have demonstrated the overall framework of the method. So here, on the green color is representing microRNA expression data, and the red color is representing gene expression data. So I hope the expression data is known to you. on the rows, these are the samples, and on the columns, these are the microRNAs. And in the red color matrix, on the rows, it is a sample, and on the columns, it is the mRNA, or you can say gene. Now, here, in biological processes, as I mentioned that in a cancer condition, or so there could be several biomarkers which get dysregulated. So their importance with respect to the disease or with respect to the cancer subtype will be variable in nature. It is, I cannot say that all the genes are responsible for this for causing the disease, or only few genes are responsible for the causing the disease. There could be indirect relationship as well, where the importance, if I quantify the importance, then it will be lower in value. So rather than implementing a feature selection method, we have developed a feature ranking, you can say feature ranking method, or feature weighing method. So here what we done, that we have ranked all the individual features. So here is one vector, you can see after the matrix, the proposed feature weighing method or approach. So this is our proposed feature weighing method. How we integrated this feature weighing method for subtype group identification, I will be demonstrating. So we developed a feature weighing method. We from the matrix, we got a vector of weights for individual biomarker, be it a microRNA, be it a mRNA, right? Now, using this information, we there is a already established method that is called as SNF, or similarity network fusion method. And then once this SNF method actually fuses or merge two different type of matrices into a single graph. Okay, so there are steps. This is an established method where we can merge the two matrices into a single graph or single matrix. Now, once we get this matrix, we can implement a clustering algorithm. Here we have used spectral clustering algorithm that will lead to different clusters that are actually denoting subtypes of clusters. Now, let's come back to our proposed approach. So here, while developing in SNF method, given two matrices, first of all, the similarity between patient. So as I mentioned on the rows, it is a patient information. So we get a patient similarity matrix for both the kind of data set, right? Now, for generating this patient matrix, so it can be simply a correlation matrix, or it can be based on a correlation matrix, or it can be a distance matrix as well. Here it is a distance matrix. So if I just remove this NWFM thing, that is multiplied by this Euclidean distance. So this is a Euclidean distance, simple Euclidean distance. So using this Euclidean distance concept, we can simply compute the patient similarity matrix. But here we have introduced, or we have given the weightage of individual biomarker from each type of data set. Okay, so the higher value will be having more impact for the distance calculation between the patient, and lower value will be having lesser impact. Now, once this matrix is generated, the similarity matrix is generated, next is converting it into graph, and then using a message passing technique, these two graphs are merged into a single graph. So now I will be demonstrating later that how it is actually done. And then once this graph or this matrix is generated, it is converted into the eigenvalue, eigenvectors are computed, and then over that eigenvectors, spectral clustering is implemented. That actually lead to identification of cancer subtype. Now, so the whole exercise or the whole study can be broken down into three steps. That is, the first one is that feature weighing approach, that is the proposed method. The feature weighing is a robust regression based approach. The second one is generating the similarity network fusion network, and then the third one is sample clustering using spectral clustering. Now, the proposed method, as I mentioned in the last slide, that the Euclidean distance, if I simply compute Euclidean distance, and if I also consider the weightage of individual biomarker, then there is a manifold improvement in the outcome as well as from the biological point of view. So first of all, in this method, we compute the mean and standard deviation, and then we compute the coefficient of variation of each of the feature. Then we develop a robust regression model, and then we fit the coefficient of variation by this way, and then we calculate the fitted covariation coefficient of variation value from the log transformed covariation value that is coming from the original data, right? And the value can be ranging from negative infinity to positive infinity. Now using the next, the weights are normalized so that it should not be having a very high impact or very low impact. So we try to fix the range, and the value is later on converted into 0 to 1 for individual weight factor. Now let's take an example how it actually does. So if I have an input data matrix, so let's say it's a gene expression data, it can be a microRNA expression data as well. So there are 6 features in the data set and 5 samples, or these samples can be patient information. All of them are cancer data. Now all of them are cancer patients. Now there is no class label, it's a unsupervised kind of analysis, you can say. Now I will be computing simply mean and standard deviation. Later I can compute so here you can see 6 features are there, so 6 data 6 points are there, right? So similarly I will be having coefficient of variation for 6 points. Next, I will be log transforming the mean as well as CV, the coefficient of variation, and then simply implementing the regression. It's a linear regression method. I will be identifying the parameters of the model, and then I can fit into the, I can identify the fitted values of the coefficient of variance. So this is the original one. This is the, so this is a fitted value, and it is a log transform fitted values. And next, then I will be finding the distance between. So here in the last step, so I am calculating the distance between the fitted value and the original log transformed value, and then it will be giving me the weight. Next, I will be normalizing it in between 0 to 1, so I will be getting the values like this. So once I get this value, so as I mentioned that I have 2 matrices. I using the weight that is computed from the previous step, that is integrated into this simple Euclidean distance calculation between the patient, and then we will be getting this sample or patient similarity matrix or distance matrix, you can say. And then the weights of the between the patient, so all these nodes are actually representing the patient information. Now the weight, the edges or the edges are the weights that can be computed in this way. So this is a part of SNF, that is established method. Next, here what happens, I will be getting 2 matrices, so sorry, 2 graphs, and then I will, using fusion iteration method here or message passing approach, I can fuse these 2 graphs later into a single network or fused network. So here actually 2 matrices are calculated. One is k nearest graph, and another one is normalized matrix, and then using those 2 information, this merging process takes place. Next step is to identify the histological subtype. So here we actually focused on histological subtypes. So once we implement the spectral clustering over this network, then we will be ending up with 2 different types of clusters that will be possibly belonging to 2 different types of cancer subtypes. Now here, the thing is that one of the students asked me that how we can do if I use a k-means clustering algorithm. So we have, there is a limitation, we need to fix the number of k here. So you can check with other sequential clustering algorithm or progressive clustering algorithm. So there you can try with that. If you don't know the number of k, how we can generate the subtypes, how many number of subtypes of clusters will be generated. So that thing is open for discussion. So this is overall approach that we have developed, and how we generated the whole concept. Next, we compared our method with some established or very popular algorithms, which are actually state of the art. So we compared our method with two types of algorithms. Now, the first type of algorithm is where so all these CC algorithms, CNF, MNF algorithm, or SNF algorithms, these are actually merging the clusters in some way or other way, and then ultimately identifying the histological subtype of cancer after implementing the spectral clustering. Right? No weightage on the biomarker is given in any of the steps in any of the algorithms. On the other hand, we also compared our proposed method with some algorithms where, for example, this WSNF method, that is a microRNA TF mRNA network based method. So it is a weighted SNF method. So in this method, this is a here the importance of biomarker is also considered while doing the analysis. So here they will be computing the importance of the biomarker, and then the importance is integrated with the SNF, and later it will, using the spectral clustering algorithm, the subtypes will be identified. Here, the major limitation is that while ranking or while giving the importance to the feature, they used microRNA transcription factor mRNA network. Now, if I'm using a network information, we all know that these kind of databases are limited in information, so they do not cover all the information that is related with the microRNA, be it a transcription factor or mRNA network. So they are, due to the lack of proper information, what happened is that we have to drop off many biomarkers from the further analysis. Also, there are several false positive information within the network. Ultimately, if I use this approach and whatever clusters I obtain and I further do the biomarker involvement analysis of the in the subtype of cancer, there is not relevant information coming up from the and this kind of approaches. Okay, so we compared our method with these types of approaches. Now, I have just shown you very limited results here. In the paper, the detailed results are provided, and please go through it. Please provide your valuable inputs to the paper so that we can modify and upgrade our algorithm later. So here, this algorithm was implemented on several 5 cancer data. Here I have only shown the result of cervical cancer. So here, first of all, we compared the performance of the approaches with respect to some standard validity indices. So for example, the average Silhouette with the accuracy, purity, normalized mutual information. So these are some weighted methods, and these are some non-weighted methods we compared our method with. And you can see that in all the cases, our method is performing better than any other algorithm with respect to any other indices. Next, we compared the math result with respect to some biological angle. So here you can see that it is a KPES, and KPES stand for CAT pathway enrichment score. So there is a so we know that when we do pathway enrichment analysis, we get there will be a group of biomarkers which are involved with certain pathways, and there will be associated p-value and FDR value with that, right? So using that p-value and FDR value, we converted that enrichment into a score, countable score. So that explanation is provided in the manuscript. So here you can, the higher the value of KPS, that means that cluster the biomarkers that are found to be associated with the particular type of subtype is highly relevant and well connected with the information. The last one is AR. AR is annotation ratio. So again, using the information of p-value and the number of genes which are involved in a particular type of biological process, using that information, we computed this annotation ratio, and higher its value, better is that cluster, or better that the more that biomarker is involved in the that particular biological process. So here I have just given you some very little glimpse of the result. A detailed and the supplementary result is provided with the manuscript. I request you all to go through the manuscript and please help me to upgrade the performance of this algorithm. The conclusion of this work is that there are some approaches available that helps in identification of cancer subtypes by integrating multiple types of information sources. These approaches give equal importance to all the features, which is not true from biological point of view. Therefore, weighing feature prior to their integration for cancer subtype identification is very important. The robust regression-based feature weighing approach used in this study helps to identify histological groups of cervical cancer patients with an accuracy of 89%. Further, on detailed analysis of differentially expressed genes and microRNAs obtained between the groups identified by different methods also shows higher biological relevance in the case of proposed method. When this method was compared with other methods that uses feature weighing approach or non-feature weighing approach, the proposed method outperformed. The method can be applied on different type of cancer data sets where identification of histological subtypes is clinically important. So this work was partially supported by the seed grant project from the IIT Jodhpur. And thank you. Please ask any question if you have. Hello?
Hello, ma'am.
Yes, yes.
Ma'am, can you highlight? Actually, I have a clarification question.
Yeah.
So, in the robust regression section, like I didn't get like what is your target variable and what is your predictors there. Can you
So
highlight that one?
yeah, so here this if I consider this mean as a x variable and this log two, so both of them are log transformed. So mean is a x variable and CV is a y variable. Later I am fitting this y variable using this using the intercept and slope information that is obtained from the regression approach, and I'm fitting it into the model. Here this is the model, and then later I'm using this information to calculate the weight. So here Y is my CV.
Okay, yeah. Thank you, ma'am.
So this is Mani here. So that was Rahul, one of my students. So I guess I had a related question. So you are trying to predict your CV using the mean. Is that what you are trying to do here? Can you please elaborate? I'm sorry I could not join from the very beginning. Maybe you may have explained it in the beginning part of your talk.
Yeah. So here Y is my CV, and then this mean is my X. So here I tried to identify that what could be the possible fitted line for CV. So that is computed using this one.
Okay, so for a well predictable feature, it will have a higher weight. Is that the
Yes,
idea?
yes, yes, yes.
Okay, and then why do you think mean, I guess certain features are more predictable of the cancer subtypes than other
Yeah.
features.
So
So,
yeah, so that's
okay.
the reason. So the higher the weight of a feature will be, the more it is would be related with the disease. So that was the idea behind why we should rank the feature rather than removing certain feature from the data.
But don't you want to use the label information there? So is the
No, there is no
label
label information. That's the thing.
okay,
So
okay,
all of them are. So that's why CV has been used as a Y variable.
okay. So you don't you just want to pick kind of the most variable ones,
Yes,
but
it's a
those
kind of
which are mm-hmm.
exactly, Professor Mani. It's a unsupervised features ranking, you can say.
Okay. So another question I had was in the two graphs that you had that you are fusing, right?
Yes, sir.
So the microRNA graph and the mRNA graph here.
Yes, yes.
If those things are consistent, I can see a way to fuse. If both of them are not consistent with each other, so
Hmm.
what does your algorithm do? Let's say
So we
microRNA
are doing
says
scaling.
that. Mhm.
Scaling can be done before implementing the steps.
No, what I have a particular question is, let's say that there are three patients which the mRNA data says that they should be in the same subtype, and the microRNA data says no, they should not be, that there is no edges between them. So in that
Yeah.
particular case, how will this algorithm resolve it? So how will it give the different weights to the different data sets?
So actually the equation is not presented here. So so for each of the matrix here, the patient matrix here, the KNN graph is generated, and it may happen. So for example, here there is a pink line present between two nodes here, but there is absence of weight or the link between here. So ultimately, uh, how much weight is there? So this quantity will be responsible for putting a edge or not to put a edge between these two nodes. Ultimately, in this graph, in, on the right hand side. So that this is an iterative process where this weight information is considered. And so I did not show two more matrices that are generated in between here. So one is the KNN graph that is generated, and then there is a normalized weight matrix. Using those two matrices, if you allow me, I will just show you the paper. Just a moment, how it is actually doing.
Sure, yeah.
Just a minute.
Yeah.
So my screen is visible.
Yes. Yeah.
So this is the paper I was talking about, similarity network fusion for aggregation data types and on a genomic scale. So here actually, the method has been described pretty well. So here the given any similarity matrix, the edges can be computed in this manner. And then from there, we will be generating a normalized weight matrix, that is P, this way. And then there is a K nearest neighbor graph that is calculated in this manner. So there is a P graph and then or matrix and S matrix. Later,
Okay.
these two, this P matrix or the normalized weight matrix is computed in this manner at the tth iteration for both. So let's say if I have two types of data types, so the first one will be updated like this, and the second one will be updated like this. The merged one or the combined one will be updated in this way. And to make it a and every iteration, it will be updated. The each individual P matrix will be updated in this manner. And if you have a more than two matrices, it's a generalized expression for merging more than two types of matrices.
Okay. 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 Thanks. Yeah.
So any further question?
I had actually one quick follow-up question is, did you find any novel subtypes based on that, or when you applied it
No,
to the
sir.
cervical cancer data, was it mapping to existing subtypes?
No, sir. So that's a very interesting thing we are also interested to know about. So actually, this spectral clustering is having this limitation. We have to fix the number of K here, and if we really want to identify some novel subtypes, we have to vary the number of K and then check that how the performance goes. So we haven't done that yet, sir. But uh, uh,
Okay.
with this spectral means if we change this spectral
Sure.
clustering
Yeah.
to
Yeah.
some
That
other
would
one,
be quite interesting to see.
yeah,
Mm-hmm.
yeah,
Yeah. So you're saying by increasing K, you can get more clusters and
yes,
real clusters. Yeah.
exactly, sir. Yes, sir. Yes, sir.
Yeah. Yeah. So please call me Mani though. Thanks.
Okay, okay.
Yeah. Yeah.